In this video, we're going to be looking at the four major types of organic molecules that we often refer to in biology as the macromolecules, and macro meaning large, and so these are some large molecules that are used in lots of biological systems. Macromolecules are large molecules that result from the joining of smaller molecules that we call monomers. So a monomer is a single unit, and monomers come together, and when you put multiple monomers together, you get a polymer. A good way to think of this is like linking cars on a train. You have one particular type of car that exists in a train, and you link that to another car, and you link that to yet another car, and still another, and eventually you get this long chain that we would call a train. Here, each individual car would constitute a monomer, single units, and then the entire train itself would be like a polymer or a macromolecule. So this is the way the major biological compounds are put together. You may recall when you were a young child, you may have had a train set like this, and you've got lots of different pieces. Not each car on the train is the same. So if you observe these different cars in this train, they're all different. But they all link together by the same process. And so you can actually rearrange the individual cars on the train and come up with several different combinations. You could put what we typically think of as the caboose. There's no reason why it really has to come at the end. You could put it further up, and whatever this particular car is, you could put it further back. And so you could rearrange these pieces and have a different train every time. And the macromolecules that we'll be looking at, the different polymers that come together and result from the joining of monomers, they can be put together in different ways. And the different ways we put them together are going to give them different shapes. And as we've talked about, when molecules have a different shape, that means they're going to have some type of different function. So let's look very quickly at some basic macromolecules and see how they're put together. The process by which we put monomers together is dehydration synthesis, and this term is just like what it sounds. Dehydration, if you dehydrate something, you are going to remove water, and synthesis means to put something together. So dehydration synthesis is literally putting something together by removing water. And we're going to use a basic example of amino acids to see how that works. So if you will recall, the basic structure of amino acid looks something like this. And we have some functional groups. We have this amine group, which is a nitrogen and two hydrogens. And then we have this carboxylic acid. And then we have what we always talk about, this varial group, or this radical group, the R group, and that can be any different number of elements, and depending on what elements you put there is going to give you a different amino acid. So what we're going to do is we're going to use a couple amino acids as an example to show how this process of dehydration synthesis actually works. Now what I have here is I have two amino acids, so here's one and here's another. And what we're going to do is we're going to put these two amino acids together. Just like we're linking two cars on a train, we're going to put them together, but we're going to do so by removing water. So what I want you to notice is we have the amine group of one amino acid, and it is next to the carboxylic acid group of the other amino acid. And that's important. They have to be in that configuration in order to react. And as we said, dehydration synthesis is going to involve the removal of water. And where we find that water is right here. Here we have a hydrogen, and here we have an OH, or a hydroxyl group. Those are going to combine, and they're going to come away from these molecules in the form of H2O. We have one hydrogen right here. We have another hydrogen here, and that's our H2. And then we have an oxygen, and that's our O. So we're going to pull those out and form water, and then this nitrogen is going to link to this carbon, and it's going to look something like this. So here we have our two amino acids, and now they are linked together. So we removed water, so this water's still hanging out around here. We took that water out and we created this bond. And if you will recall, we call that a peptide bond because it is between two amino acids. And so this is dehydration synthesis. We removed a water, and in so removing this water, we linked two molecules together. And whether it's proteins, whether it's nucleic acids or carbohydrates, they're going to link together in the same way. We're going to take away a water and put two monomers together. Now, the next thing we might be wondering is, okay, lots of biological systems involve putting molecules together, building molecules like we just did by removing water, dehydration synthesis. But a lot of biochemical processes involve splitting molecules apart. Well, in order to split any macromolecule, we're going to do the opposite of dehydration synthesis, which is we're going to add a water, so we can actually add water to this bond and break these two molecules apart, and we call that hydrolysis. And this word hydrolysis is just what it sounds. Hydro means water, and lysis means to split or to break apart. So we are adding water to this peptide bond, and by adding water, again, one hydrogen will go back to this carbon, a hydrogen and an oxygen will go back to this carbon, and one hydrogen will go back to this nitrogen. And once that happens, again, once we put this water back in, this nitrogen will take one hydrogen, and this carbon will take a hydrogen and an oxygen, and this bond will go away, and we will go back to, or we will return to having two monomers. So it would be like splitting cars on a train apart. One thing I want to do is right now I want to kind of take the time and remind us that one of the reasons that carbon is this essential molecule when it comes to organic compounds is because it has 4 valence electrons, which means it can form 4 covalent bonds. So carbon is going to be found all throughout organic compounds because it is going to be able to bond with other molecules and form long chains. So a macromolecule is a long chain. It's just like a train. And carbon is going to be necessary to form those long chains, and it's because of those 4 valence electrons. So let's look now at the 4 major types of macromolecules. First type we could talk about is carbohydrates. And examples of those would be sugars and starches. And we get those a lot in our diets from things like potatoes and breads and whatnot. Second one that we could talk about would be proteins. Proteins are molecules that we're typically going to get from eating meat and eggs and things of that nature. And proteins do a whole lot of the work in the body. They're kind of like the workhorse. They regulate chemical reactions, and they help build muscles and bones and fight diseases. Third type we could look at would be nucleic acids. And the best example of those would be DNA and RNA. And those are basically the compounds in our body that carry and withhold, store and transmit the genetic information that makes us who we are and helps us to function the way we do. And the fourth and final type would be lipids. And the examples of that would be fats, steroids, waxes. And these are the 4 major types of macromolecules, and we'll go into each of those in a little more depth in some other videos. So very briefly, let's review. There are 4 major types of what we call macromolecules. These macromolecules are polymers, and that means they are made up of monomers. So monomers, we said, were like cars on a train. We string them together, and when we string them together, we get a polymer. We said that the monomers themselves come together and connect by a process called dehydration synthesis. And dehydration synthesis is just simply the process by which we remove water. We remove a hydrogen and a hydroxyl group, and we put a bond between 2 monomers by removing water. And finally, monomers can be split by the opposite of dehydration synthesis, which is what we called hydrolysis. And hydrolysis is simply where we take water and we add it back to a bond that already exists, and the water splits into a hydrogen and a hydroxyl group, and the monomers split from one another.